This last section of chapter 13 is about how genes are switched on and switched off. In prokaryotes, like bacteria, they don't need all of their genes producing proteins all at the same time. It's either efficient or lazy, but prokaryotes, in order to conserve energy and resources, produce only the genes that are necessary for the cell to function at a particular moment in time. It would be wasteful, for example, for a bacterium to produce enzymes that are needed to make a molecule that's already available. By regulating gene expression, bacteria can respond to changes in their environment, like the presence or absence of nutrients. There are proteins that bind DNA and regulate genes by controlling transcription. Some of them help switch genes on, others switch genes off. The genes in bacteria are organized into operons, which is a group of genes that are regulated together. Usually, the genes in an operon are related to one another with respect to what they do. In the bacterium E. coli, there are 4,288 genes, and there are a cluster of 3 of those genes called the lac operon that help the bacterium E. coli break down lactose, which is the sugar that's found in milk. Here's how that works. Lactose is made up of 2 simple sugars, galactose and glucose. In order to break down lactose, the bacterium has to transport it across its cell membrane and then break that bond. These tasks are done by proteins that are coded for by the 3 genes of the lac operon. If the bacterium is in a place where lactose is the only food source, then these genes have to be transcribed so that the proteins can be produced. If it's grown on another food source, it doesn't need those proteins, so the lac genes are turned off by proteins that bind to DNA and block transcription. This is what the lac operon looks like. The first regulatory region is called a promoter. That's to the left side of the lac operon. This is where RNA polymerase can bind to begin transcription. That's the promoter right here. The other region is called the operator, which is where a DNA binding protein called the lac repressor can bind to DNA. This is the operator right here. When there's no lactose, the lac repressor, which is here, binds to the operator region, and that blocks RNA polymerase from hooking onto the DNA and starting transcription. So the binding of the repressor protein switches the operon off by preventing transcription. That repressor protein, the lac repressor protein, has a binding site for lactose. When lactose is present, it attaches to the lac repressor, changes the shape, which makes it fall off the DNA strand, the operator region. When the repressor is no longer bound, RNA polymerase can bind to the promoter and transcribe the genes of the operon. In the presence of lactose, the operon is automatically switched on. So here we have the repressor falling off. RNA polymerase can bind and start transcription. In eukaryotes, we have a little bit more complicated situation, as usual with eukaryotes. There's a TATA box. That's the sequence. Contains variants of TATA, and that's found just before a gene. in eukaryotes. This TATA box binds a protein that helps RNA polymerase get positioned on the DNA just before the beginning of a gene. So you can see what happens here. There are transcription factors, which are proteins. They bind to the TATA box and get RNA polymerase situated so that it can go ahead and start transcribing DNA and producing mRNA. Gene expression can be regulated in a lot of different ways. Like I said in that diagram on the previous slide, transcription factors regulate gene expression at the transcription level. They bind DNA sequences in regulatory regions, which are before the genes, and they can, as a result, control the expression of those genes. Why is gene regulation more complex in eukaryotes than in prokaryotes? Because these are multicellular organisms, and cell specialization requires genetic specialization. So all of the cells in our body have the same genetic material. Some of it has to be turned on, some of it has to be turned off in different cells to make those cells different from one another. So complex gene regulation is what makes cell differentiation and cell specialization possible. For years, scientists would find RNA, little small RNA molecules, only a few dozen bases long, that didn't belong to any of the three previously mentioned types of RNA. They weren't mRNA, they weren't tRNA, and they weren't rRNA. What they've discovered now is that these small RNA molecules contribute to gene regulation as well. It's called RNA interference. miRNA pieces attaches to a cluster of proteins to form what's called a silencing complex, which is what you see here. The silencing complex binds to and destroys any mRNA containing a sequence that is complementary to the miRNA, microRNA. The miRNA sticks to certain mRNA molecules, stops them from passing on their protein-making instructions. So it chops up the mRNA, translation can't happen, no protein is produced. Like I said, this is called RNA interference. So there are dicer enzymes that cut apart the larger mRNA, the larger RNA molecule, cut it into little mini microRNA pieces. Then those go out with a silencing complex, find a complementary base sequence to the microRNA, and that silencing complex's enzymes chop up the RNA into pieces, and no protein is produced. First, scientists thought that this was rare, found only in some plants and other species, but as a closer look has been taken to this, it's been shown that RNA interference is found throughout the living world, and it even plays a role in human growth and development. Researchers, by using this technique, can switch genes on and off at will by inserting double-stranded RNA into cells. The dicer enzyme cuts this RNA into microRNA, and that activates silencing complexes. These complexes block the expression of genes by producing mRNA complementary to the microRNA. The thought is that perhaps scientists can turn off the expression of genes from viruses and cancer cells, and there's a potential there to cure disease. Gene expression is also important when we look at how an organism, a multicellular organism, develops. Each of those specialized cell types found in an adult of a multicellular organism originates from the same fertilized egg cell. As an embryo develops, different sets of genes are regulated by transcription factors and repressors. And we have the differentiation thing again. Each cells of different types become unique in structure and function. Specific groups of genes control the identities of body parts in the embryo of common fruit fly. Edward B. Lewis was the scientist that found that out. He found a mutation in one of these genes actually resulted in a fly with a leg growing out of its head in place of an antenna. These genes that control position of body parts are called homeotic genes. They regulate organs that develop in specific parts of the body. Homeotic genes share this very similar 130 base DNA sequence, which is called a homeobox. These genes code for transcription factors that activate other genes that are important in development and differentiation. They're expressed in certain regions of the body, and they determine factors like the presence of wings or legs. In flies, a group of homeobox genes called Hox genes are located side by side in a single cluster on a fruit fly chromosome. Hox genes determine the identities of each segment of a fly's body. And interestingly enough, they're arranged in the exact order from head to tail on the chromosome as in the order on the actual fruit fly. So if you look at the colors here, if you look at the colors on the chromosome, we go from, if I erase it, now you can't see the colors, we go purple, green, yellow, and blue. On the fruit fly embryo, purple, green, yellow, blue. Purple is the little nose there. Green is the head, yellow is the thorax, and blue is the very end of the abdomen. So the genes are actually positioned on the chromosome in the exact anterior to posterior or head to tail order that you see on the fly. A mutation in one of these genes can completely change the organs that develop in that area of the body. Hox gene clusters have been found in the DNA of mice and in humans as well, and all of them are arranged in the same way from head to tail. See this with the mouse embryo right here to the right. Approximate body areas affected by genes of the corresponding colors. So, purple is here. That's the top of the spinal cord, the base of the brain. Green is spinal cord area. Yellow is on down farther, and blue is even farther down. So Hox genes tell the cells of the body how to differentiate as the body grows. Nearly all animals, from flies to mammals, share that same basic tool. The master control genes that control development are switches that trigger differentiation in different cells and tissues. These common patterns exist because all these genes have descended from the genes of common ancestors, which we'll explore in a couple of chapters. The environment can also play a role in gene expression. Temperature, salinity, which is salt content, and nutrient availability can all influence gene expression. We just talked about the lac operon in E. coli. It's only on when lactose is the only food source. Metamorphosis is another example of how organisms can modify gene expression. Transformations from one life stage to another, like a transformation from a tadpole to a bullfrog, is regulated by external and internal factors. As organisms move from larva to adult, their body cells differentiate to form new organs. And then old organs are lost through apoptosis or cell death. Under less than ideal conditions, like a drying pond, high density of predators, or low amounts of food, tadpoles may speed up their metamorphosis. The speed is determined by environmental changes that are translated into hormonal changes, and then that changes the cells and the rapidity, the speed at which cells change.